As you can see, this is deoxyribonucleic acid, otherwise known as DNA. It is the foundation of the diversity of the world's species. It is so long that a strand of it is about 5 feet long. But with the molecule so huge, how is it copied?
This video is an example of how DNA replicates. Deoxyribonucleic acid, otherwise known as DNA, is composed of three things: phosphates, marked by the pink clay; deoxyribose, marked by the red Twizzlers; and hydrogenic base pairs, marked by the gummy bears. Each deoxyribose, phosphate, and base are connected to each other through covalent bonds, marked by the black toothpicks, and each base pair are connected through hydrogen bonds. In the DNA, it's able to be noticed that the bases adenine, red gummy bear, and thymine, green gummy bear, always are bonded with each other through two hydrogen bonds. Likewise, cytosine, yellow gummy bear, and guanine, orange gummy bear, are always paired together, but through three hydrogen bonds rather than two.
Onwards to replication. The first step for DNA to replicate is for helicase, an enzyme marked by the red scissors, to break the hydrogen bonds connecting the base pairs in the middle of the DNA, and it unwinds the parental double helix. The area where DNA separates into two strands and the new DNA synthesis occurs is called the replication fork, and it literally looks like a fork. The leading strand builds towards the replication fork, while the lagging strand gets replicated in fragments away from the replication fork. As the hydrogen bonds are breaking because of the helicase, another enzyme called primase, which is marked by ribose, the white clay, and phosphate, the pink clay, it adds RNA primers to the DNA. But unlike DNA, RNA matches adenine with uracil, which is represented by the yellow clay, instead of thymine with two hydrogen bonds. Primase is used because it is able to start from nothing, unlike DNA polymerase, which needs something to start off.
As more of the DNA opens up because of the helicase and because primase only goes from 5' to 3' on the lagging strand, which goes 3' to 5' toward the replication. Primase forms in fragments called Okazaki fragments, as primase is not able to go backward. While both of these processes are happening, another enzyme called DNA polymerase, marked by the deoxyribose and phosphate, starts to add DNA nucleotides to the RNA primers in the 5' to 3' direction. Now, after all this is done, an enzyme called RNase H, marked by the knife, cuts all of the RNA out of the DNA. But now, because there are gaps in the lagging strand, more DNA polymerase comes and replaces the missing gaps. But now, there are gaps in the end of the leading and the lagging strands on the DNA. This part that opposes those gaps on the template DNA are parts of telomeres, meaning that they're repetitive sequences of DNA. These are cut off. Finally, an enzyme called ligase, marked by the glue stick, synthesizes the gaps in the DNA caused by the Okazaki fragments. Overall, the DNA replication process turns it a single molecule that has two DNA strands in the double helix formation and is separated into two new templates for the new DNA molecules.
Replication is an essential process because whenever a cell divides, the new daughter cells must contain the same genetic information, or DNA, as the parent cell, and this would aid organisms to be reproduced from the parents. And finally, here's our sources, and thanks for listening.